Assalamu alaikum, my dear students. Today we will be talking about viruses. First of all, the meaning of the term virus. Virus is a Latin term, the meaning of which is poisonous fluid. So, viruses are, in literal terms, they employ a poisonous fluid. Viruses are basically submicroscopic, or you can say ultra microscopic, infectious agents which are capable of infecting plants, animals, bacteria. Okay, and these submicroscopic infectious agents, they are inert outside the living host cells, but when they enter into the host cells, they become active. Okay, outside the living cells, they are as good as chemicals, but when they enter the living cells, they become active. First of all, we will discuss the history of the field of virology. Okay, how the subject of virology came into existence. First of all, the term virus, it was coined by Louis Pasteur, who is regarded as father of modern microbiology. The term virus was coined by Louis Pasteur, who is also regarded as father of modern microbiology. The study of viruses is called virology. It is sub branch of microbiology. This is taken as a sub branch of microbiology. Then Wendell Meredith Stanley, who also contributed a lot in the field of virology, he is regarded as father of virology. Okay, these are important terms, you have to remember these terms. While Louis Pasteur is regarded as father of microbiology, Stanley is specifically regarded as father of virology. Then Edward Jenner in 1796, he developed first successful vaccine against smallpox. Edward Jenner in 1796 developed the very first successful vaccine against smallpox. Then Louis Pasteur in 1880 discovered infectious nature of rabies and developed anti-rabies vaccine. Then in the year 1886, Adolf Meyer discovered tobacco mosaic virus, or you can say tobacco mosaic disease. Okay, this is a disease caused in tobacco plants by tobacco mosaic virus. Dmitry Ivanovsky in 1892, he extracted the juice from mosaic infected leaves of tobacco, then passed this juice through bacteria proof filters, Chamberlain filter. The juice he got, the extract that he got after filtration, it was still effective. That means it can still cause disease in the healthy plants. He concluded that the extract contains the toxin. The extract contains the toxin produced by the bacteria, because at that time there was no concept of viruses. So Ivanovsky, he extracted the juice from mosaic infected leaves of tobacco. Then this juice was after then filtered through the bacteria proof filters, and this juice was still able to cause infection in the healthy plants, and he concluded that it is actually the toxin produced by the bacteria that enters the bacteria proof filters. But later on, Martinus Bijernik, he repeated the same experiment and found that the new infectious agents in the juice, he called it Contagium vivum fluidum, soluble living germ. Okay, and reintroduced the term virus originally proposed by Louis Pasteur. Bijernik, he repeated the experiment and he found that the new infectious agents in the juice, They are not the bacterial toxins. Okay, they are not the bacterial toxins. He named them Contagium vivum fluidum, soluble living germ. Okay, soluble living germs. Sorry, and he reintroduced the concept of viruses, which was originally given by Louis Pasteur. Okay, so one of the key was of the view that the extract contains the bacterial toxins, but Bejernik, he after repeating the same experiment, he named the toxins as Contagium vivum fluidum, which in other words means virus. Then W.M. Stanley in 1935 he purified and crystallized the tobacco mosaic virus for the first time, and for this work he was awarded Nobel Prize in the year 1946. This was all about the history of the field of microbiology and virology. Now some important characters, or you can say general characters of viruses. Viruses are ultra microscopic, or you can say submicroscopic. Their size ranges between 10 to 400 nanometer in diameter, and they are seen by electron microscopes. Okay, they can only be seen by electron microscopic. These are ultra microscopic infectious agents whose size ranges between 10 to 400 nanometer, and these agents they can pass through bacteria proof filters. Because their size is smaller than bacteria, they can easily go through bacteria proof filters like Chamberlain filters. These agents they possess only one type of nucleic acids. Either they contain DNA or RNA, not both. This is very much important. A virus contains either DNA or RNA. They do not contain both DNA and RNA. These agents they are acellular, and hence they can be crystallized. They don't have cell membranes. They don't have subcellular organelles. They don't possess mitochondria. They don't possess other structures which are present in the bacterial cells and eukaryotic cells. Okay, they are simply the DNA and the protein coat, or RNA and the protein coat, and hence they can be crystallized. Okay, they are acellular. They don't have any cells. They produce characteristic symptoms only in host plants. These agents, when they infect the host plants or host organisms, be it animals, plants, they produce characteristic symptoms. They are obligate intracellular parasites. That means they are only able to live when they get an entry into the living host cells. They cannot be grown on artificial medium. Okay, they can't be grown on artificial medium. They are only able to live once they enter into the living cells. That is why they are known as obligate intracellular parasites. They lack any functional autonomy. That means they don't have any machinery, okay, of their own. Instead, they use the machinery of the host cells to manufacture their DNA, RNA, and proteins. They lack pigments. They don't have any locomotory organs. Okay, they don't show locomotion. They don't have any organs which will assist them in movement, and they are also lack sex organs. They synthesize their proteins on host ribosomes. They use the host ribosomes. Okay, inside the host cells, they use the host ribosomes to manufacture their proteins. They are not affected by antibiotics. They produce new strains by mutations. Okay, these agents they cannot be affected by antibiotics because antibiotics are specifically effective against bacteria. Okay, these agents they are not degraded. They are not affected by antibiotics. Okay, they respond to heat and stimulus and can be inactivated by chemicals or heat. Okay, they respond to heat and other stimuli, and these agents can be inactivated by chemicals Okay, they can be crystallized, they can be degraded by some chemicals, and they can also be deactivated by heat. They possess two components. The first one is nucleic acid, and the second one is protein. These components, they get synthesized and assembled within the host. Okay, both nucleic acids and the proteins, they get assembled, they get manufactured, and then later on assembled into a viral particle inside the host cell. The viruses are still one of the most puzzling experience. Okay, I can say viruses are the biologist's puzzle. They show both living as well as non-living characters. That's why they have puzzled the biologists. Okay, their nature is not so much easily comprehended. You cannot easily comprehend the nature of viruses, because at one point of time they are just like non-living things. Okay, they show non-living characters, and once they get entry into the living cells, they become living. Okay, that is why they are regarded as the borderline of life and death. Okay, they are the twilights of life and death. Okay, so the living characters and non-living characters of viruses, we will try to compare uh, the living characters and non-living characters of viruses. First of all, the viruses, they possess the genetic material. Okay, and this is the characteristic of living organisms. Only living organisms possess genetic material. Okay, they can be they can multiply to increase their number and also undergo mutation. This is also the characteristic of living organisms, because only living organisms can increase their number, and they have only the living organisms have the ability to undergo mutation because they possess genetic material. Okay, it is genetic material that undergoes mutation. The third one is they respond to external stimuli such as radiations, chemicals, and heat. This is also the characteristic feature of living organisms, because only living organisms, they respond to stimuli, external stimuli such as radiations, chemicals, and heat. They exhibit host specificity. That means a specific virus infects a specific host. This is also the characteristic feature of living organisms. And they are capable of causing a plethora of diseases in plants, animals, and bacteria. Okay, this is also the characteristic feature of living organisms. Now, the non-living characters of viruses, they can be crystallized like non-living objects. They can be crystallized like chemicals. Okay, because since chemicals are not living. Okay, same is the case with viruses. You can crystallize them, and that makes them non-living. Okay, they are considered as non-living because they possess they can be crystallized like chemicals. The second non-living characteristic feature of viruses is they are like inert chemicals outside the host. जैसे कि मैंने आपको पहले भी बोला, outside the living host they are as good as inert chemicals, but once they enter the host cells they become active. The third one is they lack cell membrane and other cell organelles. That means they are acellular. They don't have any cell membrane. Cell membrane is the characteristic feature of living organisms. They don't possess any intracellular, or you can say cell organelles. Cell organelles includes mitochondria, ribosomes, all these features, all these intracellular cell organelles, they are absent in viruses. They lack functional autonomy. That means they cannot perform their own functions. Okay, they don't have any machinery to synthesize their proteins, to synthesize their DNA, to synthesize their RNA. Instead, they use the machinery of the host cells to get their proteins and DNA manufactured. That means they don't have any sort of functional autonomy. And the viruses, they don't show any metabolic activity. There is no metabolism whatsoever present in viruses. So these are some important characteristics of viruses which or shared between living and non-living things. Okay, so this is all for today. In the next part we are going to talk about the structure of viruses and other details. Till then, stay blessed and thank you all.